Thank you very much, Enrique and Dennis, for the invitation. So, yes, so I am a group leader in Newcastle University for about 1 year already. And the work I'm presenting today is in collaboration with the group of Alfonso Valencia and Barcelona Supercomputing Center. So actually, the work that we uploaded to bioRxiv this summer is the PhD thesis of Maria Rico, who's been co-supervised by Alfonso Valencia and myself, and also with the contribution of David Juan from Pompeu Fabra University at Barcelona as well. So a little bit of introduction about CMVs. So the majority of human diversity studies normally focus on single nucleotide variants, but it's important to take into account that copy number variants, or CMVs, contribute significantly have contributed significantly to hominid evolution, adaptation, and many diseases. And actually, it's estimated that around 12%, maybe 50% of the human genome is subject to copy number variation when we compare different individuals in different populations. So what do we mean by copy number variation? So if you imagine that here you're seeing two chromosomes. So one chromosome will be the parental copy and the copy from the mother and from the father. Normally, in the majority of the genome, we will have two copies, one in one chromosome and the other one in the other chromosome. But in some regions, copy number variable regions, we may have extra copies. So maybe we have an additional copy here, so we'll have a copy number of 3, but the extra copy can be like in the same chromosome or anywhere else in the genome, or we can have losses. So when we lose one copy, we talk about hemizygotic deletions, and if we lose both copies, both alleles, then we'll have 0 copies of that region in that individual, and that would be an homozygotic deletion. I would like to also differentiate between germline and somatic CMVs. So germline CMVs would be CMVs that will be present in all the cells, in most of the cells of one organism, so one individual, and the variability refers to differences in the population, so differences among individuals. You may also hear about somatic CMVs, or sometimes called copy number alterations or copy number aberrations, and these are differences within an individual. So certain tissues with age tend to accumulate copy number variation, and in particular, cancer cells also are very well known to accumulate copy number changes. But the work I'm presenting today is focusing in population variability in copy number variants, or germline CMVs. And the objectives of this work were first to perform a comprehensive comparison of 5 different CMV maps. And with this data, we wanted to evaluate specifically the impact of copy number variants on protein coding genes. So I will present the differential effect of CMVs in coding and non-coding regions of genes, so exons and introns, their differential effect depending on the evolutionary age of the genes, and finally some data about how CMVs in genes can be associated with gene expression changes. The material we have used, as I said, are 5 different CMV maps. All of them appeared in 2015. That was a year particularly active in the CMV field. So first, we got the CMV map from Sarai et al., which basically is a meta-analysis of the data that was available at the time in the database of genomic variants. Then we also had the maps by Hansacker et al. And Avisov et al. So both studies are using data from the phase 1 of the 1000 Genomes project, but they use different algorithms and analytical strategies. So the one by Hansacker tend to be detecting mainly large CMVs, and in the case of Avisov et al., it's mainly about small deletions. And then later that year in summer, we had another paper by Sudman et al. In Science. And in this paper, they tried to characterize copy number variation all over the world. So they sampled human genomes in very different locations, in different continents, like trying to expand the variability that is incorporated in the 1000 Genomes Project. And finally, by Sudman et al. In Nature, there was the final map of the 1000 Genomes Project for more than 2000 humans. So all of these maps are, we think they are complementary, and they all, at the end of the day, have the common goal, that is to try to reflect a part of the human variability, the structural variability. Okay, so as I said before, we are particularly interested in evaluating the impact of CMBs in genes. So if you think about how a CMB can impact a gene, there are at least three basic scenarios. So we can have CMBs that expand entirely, incorporating a whole gene, sometimes even more. So these kind of CMBs will result in duplications or deletions of the entire genes. We can also have exonic CMBs. So by exonic CMBs, we mean CMBs that they are not affecting genes entirely, but affect the copy number of exons. And then finally, we are also interested in looking at the intronic CMBs, that would be copy number changes that affect the introns without affecting the coding sequence of protein coding genes. So we look at the number of these different events in the different maps, and we differentiate it between gains, losses, and gain losses. So gain losses would be regions that sometimes can be gained, sometimes can be lost in different individuals. So what you can see is that the number of events is very different in its map, and the most recent map, most comprehensive map, by the final phase 3 map presented in Sudman et al. In Nature, the 1000 Genomes Project, is the one reporting the highest number of events. Interestingly, for whole gene CMBs, we see many gains in most of the maps, except Avisov, which is actually only reporting deletions. And interestingly, in the case of intronic CMBs, we found that the majority of these events tend to be deletions. So we got particularly interested in the fact that the most abundant form of copy number variation affecting genes is actually intronic CMBs. You can see, you can compare the numbers in the y-axis. So we decided from here to characterize further the intronic deletions in genes. So in a previous work published in 2013, we observed that whole gene CMB gains tend to be significantly enriched in evolutionary young genes, where we also correlated the data with DNA replication, and we found that late replicated regions tend to accumulate evolutionary young genes that tend to be, in turn, copy number variable in the population. So we wonder if these intronic deletions were equally affecting all and young genes in this case, because at that time we didn't have enough resolution to distinguish intronic versus exonic or whole gene CMBs. So to date the age of genes, what we did is we took all the human genes and all the different additional species available in Ensemble to try to see to detect the ancestral species where the gene first appeared. So imagine that we have one gene in human, in Homo sapiens. We can detect autologous genes in chimpanzee, orangutan, macaque, and so on. So finally, we don't find we don't find this gene in mouse and other most distant species. Then we can date the birth event at the ancestor of primates, the simiformous ancestor, and that would be the gene age. So we have done this for more than 18,000 human genes. We were able to date them using this gene family trees from Ensemble, Compara, and this would be the different gene ages that we were able to group the genes into. So it is interesting for many reasons to classify the genes in different gene ages. One of them is that functionally they tend to be very different. So ancient genes are enriched in housekeeping genes and genes with essential functions, while in the case of young genes, they tend to be enriched in tissue-specific or cell-type-specific functions. So with that, what we then did was to calculate the proportion of CMV genes or genes showing deletions for each gene age class. Okay, so here what I'm showing is the percentage, for example, for fungi-metazoa, that are the older genes we have. This would be the proportion of the genes that show deletions affecting their coding sequence, either by whole gene deletions or exonic deletions. What you can see is that the proportion of having deletions in the old genes is lower than expected by chance. That is what is indicated by these black asterisks. While in the case of younger genes, they show a proportion that is higher than expected by chance. So similarly to what we observed in the past for CMV gains, we can see that also deletions in coding regions tend to be impoverished in old genes and enriched in evolutionary young genes, where you actually expect to see more polymorphisms in general and more variability in the population. The big surprise was that when we did the same analysis with intronic deletions, what we found was the opposite. So basically now the intronic deletions tend to accumulate in the older genes, and this accumulation is highly significant. And as you can see, the patterns are very different with the 3 maps that have highest resolution to detect deletions. So both in SUDMAP Nature, Surrey analysis, and the Abyssal maps, we see very similar patterns. So suggesting that this intronic variation is affecting all genes, and many of them that we check that have essential functions that still show variability within introns. Okay, so this figure, this final panel of figure 2 is summarizing all these results. So we take the percentage of deleted genes per gene age. So we take just the genes that show deletions per each of the evolutionary age classes. What you can see is that older genes tend to have mainly intronic deletions, but in the case of younger genes, those genes born during primates evolution, is more frequent to have actually whole gene deletions than to have intronic deletion. So it's a very different pattern depending on the evolutionary age. Okay, so we have different intron sizes in the population. So actually what this means is that we have different size of genes for many genes in different individuals. Because if we just consider this schematic, this gene, here in red would be 2 regions that can be deleted. So if you delete one of them or the 2 of them, you may have a shortening, a reduction in the size of the actual gene. And we wonder what consequences this may have, if any, in the population. So we characterize, this is summarized in figure 3 a little bit, what's the proportion of the intron that can be lost. And you can see that there are introns that more than 50% of the intron can be lost here in panel A. And if you see panel C, this will be the effective change in gene length by intronic losses. So some genes may lose up to 10 or even 20% of their size because of intronic variation. And here in the panel D, we have one of the most extreme examples, so the SLC1A gene. What you can see is that there is this huge region in this intron that when it's lost, there is a very significant reduction in the gene size with more than 30 kilobases less in the individuals that are losing this intronic sequence. Okay, so we finally wonder, so can we detect that any gene expression differences associated with this intronic variation? So to answer this question, we took advantage of the RNA-seq data available from the UBADIS project. In this project, they generated expression data for more than 400 individuals that were also sequenced as part of the 1,000 genomes project. So we have individuals with much copy number information and gene expression data. And so what we did is first we look at we tried to see in this subset of individuals how many cases we had for each of the 3 types of deletions. So we could find 51 whole gene deletions in at least 2 individuals. We also found more than 200 exonic deletions, and we found almost 500 intronic deletions, so purely intronic deletions. So what we did here is to do a differential gene expression analysis. So for each gene, we compare the expression levels in the people with the deletion compared with the people without the deletion, like the wild type versus deletion, if you wish. And also we only considered cases where the only 2 possible cases were like the normal state, 2 copies, or 1 copy, hemizygotic deletion. And then on the bottom figure, you can see the actual number of differential expressed genes. We found about 15 genes that are differential expressed when you have whole gene deletions, about 30 with exonic deletions, and about 52 with intronic deletions. So to see if these numbers were more than expected by chance, we repeated all the differential analysis for every gene, but now randomizing the class levels of the donors. So instead of taking the real people with the deletion and comparing them against the rest, we randomly took the same number of individuals in the population and performed the test again 10,000 times. With that, we have in this other bar plots in white with these error bars, we have the average number in these random permutations and the standard deviation, and you can see that in the 3 cases, the number of genes differentially expressed is higher than expected by chance. And given that intronic deletions is the most abundant form of copy number variation affecting genes, we think that this potentially could have some impact in the future interpretation of intronic variation. We think that it hasn't been explored too much until now. Okay, so with that, I would like to wrap up to my final slide. So in figure 5 of the article, we try to summarize all the information. And I think the main message of this work is that copy number variation will affect differentially ancient and young genes. So in ancient genes that tend to be enriched in essential functions, we will mainly have intronic variability, and this intronic variability may affect the expression timing, expression levels, and we also have actually some preliminary new results that are not included yet in the paper. The next version of the preprint will incorporate them that suggest that also may be affecting splicing as well, and that these kind of things will be happening, as I said, in evolutionary ancient genes. But then in evolutionary young genes, the copy number variation in coding regions will be more frequent, and this is probably accounting for a lot of gene birth, so the birth of new genes by duplication, also the death of genes that tend to be eliminated also more often, and maybe also reshaping of proteins by partial exonic CMPs as well. Finally, I would like to thank all the people that have made this work possible. Principally, Maria Rigau, a very talented PhD student who actually has done all the analysis that I have presented today and contributed with many ideas. She's co-supervised by Alfonso Valencia, and actually we started this work when I was still in the group of Alfonso, who now is working at the Barcelona Supercomputing Center, and also the co-author David Juan, now in a postdoc in Universitat Pompeu Fabra in Barcelona as well. And finally, the funding I have from the Wellcome Trust and the Obra Social La Caixa funding for the PhD for Maria Rigau. With that, yeah, I would like again thank Dennis and all the organizers of the online club. Thank you very much for your attention.